Hello and welcome to the third webinar in the Organic Seed Production 6-webinar series organized by the Organic Seed Alliance and the Multinational Exchange for Sustainable Agriculture. We're hosting one webinar in this series on the third Tuesday of each month through November 2016, and you're welcome to attend as many of them as you'd like. This is your host, Alice Formiga from the eOrganic Community of Practice. Today's presentation will last about an hour, and then we'll have 30 minutes for your questions. So today, we welcome back Jared Zystro of the Organic Seed Alliance. Here's a picture. And Jared has been presenting all the webinars in this series so far. And today, we're also very glad to introduce Shannon Carmody of Washington State University. And today they're going to focus on the management of diseases and pests in organic seed crops. So with that very brief introduction, I'm going to pass over the screen control and the microphone to Jared Zystro.
Thank you so much, Alice. And I'm glad to everyone who was able to attend today. I think this is a really important topic because if we're going to do a good job, if you're considering growing seeds either for your own on-farm use or for commercial sale, understanding what diseases are important and which ones are seed-borne and how to manage them is going to be a really critical skill for you to develop during your growth as a seed grower. So what we're going to be talking about today, recognizing that there are people listening to this webinar all throughout the country and even throughout the world, and people who are working with many different crops, we're not going to be able to go through in detail on a crop-by-crop basis and talk about what diseases there are. Instead, we're hoping that we can give you the tools that you need to understand big picture, how do you manage environmental conditions and make good choices to reduce your risk of developing problematic outbreaks of diseases and pests in your seed crops, and how do you find out what diseases you may have and how do you treat those diseases if they are seed-borne. How do you treat your seeds? So I'm going to start by talking more about the management in the field of diseases and pests, and then Shannon will cover more around testing and treatment of the seeds. So this is my basic outline that I'm going to be covering in the next 20 minutes. And we start by talking about how to consider which pathogens may be potentially problematic for your seed crop. And then I'm going to be talking about how to reduce opportunities for those pathogens to infect your fields. I'm going to be talking about how to choose appropriate genetics to reduce your risks of seed diseases, and then talk about how to manage environmental conditions to reduce the ability for those diseases to thrive in your fields. And finally, what to do when those diseases do appear. What are some management tools that you might consider? So first, how do you consider what diseases are important? Crop diseases and opportunistic organisms are a regular part of life for most farmers and seed growers. However, you may not be able to manage, and it may not make sense to aggressively manage all of the pathogens that could be present in your field. So what you want to think about is, first, which diseases are extremely virulent, which ones are likely to spread and to significantly damage either the quality or yield of your crop, either damage your seed yield or subsequently damage the quality or yield of the crops that are going to be grown from the seeds you're producing. Second, are those diseases seed-borne? Many diseases that can cause significant problems for you are either transmitted through the air or through the soil and don't necessarily, or vectored by insects, and don't necessarily get carried from infected seed into other fields. So knowing which diseases are seed-borne is really an important thing to understand, because those are going to be the ones, if they're highly virulent and they're highly seed-borne, those are diseases that you need to be aware of and alert about and actively avoiding and managing. Finally, there may be diseases, as you're looking through the potential diseases that are problematic for your crop, that just aren't found in your region because of the climate is not conducive to them, or just because they haven't yet made it to your part of the world. So how do you find that list of potential diseases? One of the best resources out there is talking to plant pathologists like Shannon, or most states will have extension pathologists that are there to consult with farmers. There are some specific seed pathology programs like that up at Washington State that can help you as a seed grower, but many state extension offices have pathologists that have a strong general knowledge of diseases that are particular to crops in your state and in your area. There are certain references that can be helpful. The American Phytopathological Society produces a series of crop disease compendia that may be worth investing in depending on the crops that you're growing, and those have great color pictures and diagnostic tools that can help you. There are a few seed-specific references. Organic Seed Alliance's publication that we did in conjunction with Seed Savers Exchange, the Seed Garden, has a table of some of the most common seed-borne diseases on it, as well as John Navazdio's Organic Seed Grower book has a table at the end that lists those. Many of Organic Seed Alliance's crop-specific publications do list diseases for those crops in the publication. So once you have an idea of what some of the potentially problematic diseases you could be facing are, that gives you an opportunity to better understand those diseases in terms of what is their life cycle, what are some of their climatic preferences, what alternate hosts might they be able to survive on, what are some controls that are available to limit their growth and spread. So the more that you can arm yourself with that knowledge ahead of time, the better prepared you will be to be proactive in controlling these diseases in the field. The first step in successfully managing diseases and pests is before you ever get to growing the plants. So one of the most important aspects of pest and disease management in organic farming is good rotations. So by rotation, this means generally means a prolonged period where you avoid planting crops from the same family in the same location for several years. There's the length of that rotation will depend on what the crop is, what diseases you're trying to manage, because essentially what you're trying to do is give enough time for diseased spores or pest eggs to eventually lose their viability before you come back in to plant again. So I know that, as far as the timing of it, many farmers look at rotations on a 3 or 4 year basis. I saw Alex Stone at Oregon State University had been working with long-term successful organic farmers and found that their one of their #1 challenges over the long run had been managing diseases, and that many of them had found that moving up to something more along the lines of a 7 or even 8-year rotation, meaning that they weren't planting the same family of crops in the same location for up to 8 years, that was really what became necessary for managing some of the most problematic diseases in their crops. So as you can see, something like that will often require you to have a certain size land base so that you can manage these long rotations before you come back and plant the same crop again. It will also involve strategies around your market plan so that you can be successfully growing and marketing crops from several different types of plant families, so that you're not forced to just grow 1 or 2 families again and again. So after rotations, residue management is often another important thing, and this is something that to some extent comes in conflict, I feel like, with goals of reduced tillage and limiting the leaving as much organic matter on the surface of the soil to prevent runoff. So this is something that may require a certain amount of decision making as far as a compromise between reducing tillage and leaving organic matter on the soil versus managing disease. So this is something you'll just need to consider based on what disease and diseases you're trying to manage, whether or not you want to be actively incorporating the residue back into the soil, breaking it down so that the soil microorganisms can eliminate all of that crop residue in a rapid enough fashion before you are planting again. So again, that will depend on your specific diseases that you're managing and kind of your larger management plan. Finally, managing alternate hosts and volunteer plants. Many diseases that infect your seed crops can also infect other plants, either of that same family or even of different families, and those plants may be other crop plants on your farm or weeds that are common on your farm, and so understanding what those alternate hosts might be and managing them either by keeping your field edges clean of weeds that can be potential hosts for the diseases that can infect your field. And also for many seed crops, what you'll find is that in the process of harvesting your seed crop, there's a certain amount of shattering that happens. Seeds are dropped on the ground, and then you end up with volunteer plants from your seed crop the subsequent year. So both for the goal of reducing risk of accidental outcrossing by having volunteer crops growing with unintendedly, and also reducing opportunities to harbor the pathogens, you want to be managing and eliminating volunteer plants from previous seed crops. Shannon may talk about this concept later, but an important thing that thinking about when we're talking about all of these things about rotations, residue management, alternate hosts and volunteers is this goal of eliminating what's known as a green bridge. So many pathogens will need some living plants to be able to continue to survive, and if you can in some way have at some point in your season a long enough gap where there are no living hosts for that pathogen, then you can help disrupt the pathogen's life cycle, as opposed to if you have kind of a constant rotation and of the same of potential hosts that exist on your farm, then the pathogen can survive throughout the year and continue to grow into the next season. So another place that diseases can find a foothold. One of the most important things to be aware of is being conscious of seed-borne diseases as they come onto your farm. So understanding what some of the most prominent and problematic seed-borne diseases are, and making sure that you're not bringing those in as you're buying or sharing seed with other seed growers or from other sources. So if there are problematic seed-borne diseases, find out if the person that you bought the seed from has tested those seeds for important seed-borne diseases. Make sure that the tests come back negative. Many growers will proactively treat their seeds with hot water treatment or other treatments like that Shannon will be discussing later before planting. So just as an added precaution about importing seed-borne diseases onto their farm. And then another place that diseases can persist is on if you're reusing and recycling seedling trays in the greenhouse, having some sort of protocol for keeping your greenhouse clean, sterilizing your seedling trays, and to reduce the opportunity for pathogens to get in at the seedling stage. One tool that is available depending on sort of how you're choosing crops to grow for seed is looking for seed crops that have some degree of genetic resistance to some of the most problematic diseases in your region. So many many catalogs and varieties will have listed what diseases they are resistant to, either resistant through a qualitative strong single gene resistance that may have a name or an acronym associated with that particular resistance, or some crops maybe have a more what is known as horizontal or durable resistance. That's a function of many genes that help provide resistance, either through something physiologic, like maybe having additional spines on the plants that keep the insects from feeding, or thicker leaf tissue that prevents infection or outbreaks of diseases. But choosing varieties based on their ability to resist diseases is often a useful tool, and something that you can consider as an opportunity to manage diseases on your farm, both in terms of choosing seed crops, but also say you did have a variety that you really wanted to grow that was part of your business plan or your farm plan, but it was susceptible. If you have reduced the amount of pathogens growing on your farm by growing resistant variety several years, then you may find that you have a window to grow something that's susceptible for a year because you have such a low amount of pathogen reserves on your farm that you can take an opportunity. So thinking about using resistant varieties in your rotation to help keep the pathogen pressures low, even if you do intend to grow susceptible varieties occasionally. Once you've thought about how you've managed your farm before you've planted and you've chosen good varieties, then at planting and after planting, some of the ways that you can manage your environment to reduce the ability for diseases to grow and spread. First and foremost is the idea of just good basic crop practices that keep your plants happy, so making sure that they're not water limited or nutrient limited, that they have the ability to have uninterrupted healthy growth throughout the season. Stress plants are due to abiotic stresses like drought and nutrient deficiencies, can be more susceptible and attractive to pests and diseases. And so by controlling the environment to keep your plants growing well, you can reduce their susceptibility that way. Another thing that you can think about is when you're talking about the timing of your planting, if you have some room for planting, and this goes back to, I think, the very first webinar that we had in this series, where working backwards from your crop harvest time and from your ideal pollination dates to understand, how much room do you have during planting. If you have some room to choose a planting date, you can think about whether it makes sense to plant earlier or later in the season to avoid and reduce environmental conditions that allow those pathogens to thrive. So if it's a disease that thrives in cool, wet conditions, can you plant that crop a little bit later in the year so that it's already beginning to warm and dry up, for example, before so that the pathogen has very limited time period where there's the crop in the ground and the conditions are good. Likewise, if it's a pathogen that really thrives in the heat, can you, look at potentially overwintering your seed crop and avoiding the hot times, or planting it earlier so that the plant is close to finishing up before the disease conditions are really optimal. Another tool that you can have is thinking about airflow. Having good airflow and relatively dry conditions around the plants helps reduce many problems with pathogens. So one of the tools for that is having wide spacing both within your rows and between your rows. Most seed crops require larger, wider spacing than growing those crops for vegetables. So being aware of how much space the seed crops will require and providing that space up front so that there's always airflow passing through the rows can be really important. Using stakes to hold your plants upright and keep them from filling up the entire space between the rows is an option sometimes. And then row orientation, so thinking about the direction of prevailing wind so that you can orient your rows to encourage wind to flow through the rows and reduce the humidity that's being formed as your plants are transpiring and adding water vapor into the air. If you can keep having wind blowing through there, it can carry that water vapor away and keep the conditions relatively dry. I think maybe Shannon will speak to this. I believe Lindsay DeToy, who's the Washington State seed pathologist, did an interesting study looking at the power of row orientation and controlling some diseases in brassicas, and found that by careful selection of the way that you orient your rows, that you can have significant impacts in reducing diseases on your plants. Another important tool is thinking about watering. So as we've talked about in previous webinars, avoiding as much as possible overhead watering, especially as the crops get larger and begin to flower. By switching over to drip or furrow irrigation, you can keep the leaves from getting wet, keep the flowers from getting wet, keep the fruits from getting wet, and that so many times will, because many pathogens, especially fungal pathogens, will tend to thrive in moist conditions. By keeping water off the plants, you're going to be able to reduce risks that way. If you do need to water overhead, think about the timing of that watering. So watering at a time when the plants can dry quickly. So this is often a compromise where watering at night may be more efficient because you have less evaporation occurring. But if you water early in the evening, then the plant leaves would be staying wet all night and giving a longer window for the pathogens to grow. So often, sometime early in the morning can be a good time to consider watering if you're watering overhead, so that you're watering before the sun is out, or the sun is, before it's getting too hot, to reduce the risks of evaporation and getting more efficiency from your watering. But then fairly quickly after you're done watering, the sun will be up and drying the plants, so that there is a relatively short window of time when the plants are wet. Likewise important is if the plants are wet, either from watering or because of morning dew or rain, that's a time to, if at all possible, avoid working in the field, because you want to avoid injuring the plants. You want to avoid spreading potentially kind of contaminated water between the plants. The water at this point may have spores in it that you can spread from a diseased plant to a still healthy plant with through your wet clothes and your in your hands. So letting the plants dry before you work is an important tool. So at this point, most of what I've talked about is basic sort of preventative strategies. What do you see? What should you do once diseases are starting to appear? Again, this is going to depend on what the diseases are and what point in the life cycle they're occurring. If it's if it's not a seed-borne pathogen, if it's not extremely virulent and it's kind of late in the life cycle, it may not really be a concern. You may be at the point where the plants have already are close to maturity and going to be able to set seed and yield well without any active management. But earlier in the season, some of the things you want to be doing is, as you're seeing individual plants that are infected, both you want to remove those plants and take them far away from the field and destroy them in an appropriate fashion to keep those pathogens from being able to persist on your farm. So this has two benefits. One is it's taking those infected plants out of your field so they don't, it makes it harder for the inoculum that's being produced on those plants to spread to the still healthy plants. It also is important as a seed grower to be roguing out and removing plants that are susceptible, because it's going to improve the genetics of your seed crop to take out the ones that are most susceptible, so that they don't contribute to the subsequent year's seed and subsequent generations' genetics. You want to, there are many OMRI-approved controls, so so these are things, fungicides and things that can control bacterial diseases that are approved for use in certified organic operations. So understanding what those are and how to use them is going to depend on where you are and what crop you're growing. And sort of general advice with that is that many times they work best when you apply them relatively early in the process of an outbreak. So, as soon as you're seeing any signs, then it would be a good idea to apply those controls. There are many sort of more biological controls that, using beneficial organisms that you may be really wanting to apply even before any signs of outbreak appear. But you're generally going to want to, most of the organic controls require you to be fairly proactive and apply them early in the disease outbreak, and they may have more limited ability to be able to overcome a full-blown case of disease once it's progressed into later stages. So understanding what those are and how to apply them is going to, again, depend on what crops you're working with. Finally, unfortunately, sometimes you come to a situation when the disease is problematic enough that you really need to make the call to just destroy the field. And this is something that often, if you're growing on contract for a seed company, you want to be having conversations about disease actually really from the get-go, even from when you're first getting the contract. Your seed company representative can help you understand what diseases might be problematic on this seed crop, whether or not the variety you're growing could be susceptible, any recommended controls. So having that conversation early is important, but you definitely want to have a conversation with the seed company as a disease is progressing. If it's something that is seed-borne, the seed company may request that you destroy that crop because what you don't want to be doing is bringing in more infected seed into the seed marketplace so that it can spread to other locations. So understand that sometimes this is a choice that you're going to need to make, and carefully consider it, but know that it is not uncommon to need to just remove that particular crop from the field. Briefly, another tool that is important is thinking about after you harvest. So when you've harvested your seed, you're not necessarily out of the woods yet in terms of diseases and pests. Seed-borne diseases and pests can find their way into seeds and storage, or there can be a small amount of inoculum that's present on the seeds themselves or on some of the chaff that's in with the seeds. And so what you would want to do after you've harvested your seed is first, as quickly as possible, clean your seeds as much as possible, because seeds are much more dense and less porous than the chaff that they're going to be with when you do the initial harvesting. So the cleaner you can get it, the less chaff that you have in with your seeds, the less opportunity for pathogens to grow in your stored seed. And then second, dry seed is having fairly well-dried seed is a good tool for reducing the risks of outbreaks in your stored seed. So many, both pests and both storage pests and diseases won't really spread and grow if your seed is dry enough. And so getting your seed well dried before you put it into storage is a really important part of your overall toolbox. So I think we'll be taking questions at the end, but at this point I want to pass it over to Shannon Carmody, who works as a research assistant in the Washington State University Vegetable Seed Pathology Lab.
Thanks, Jared. As you said, my name's Shannon. I work in Dr. Lindsay DeToy's seed pathology lab here in Mount Vernon, and I got into pathology, seed pathology, I should say, after becoming involved with seed, and I grew my first commercial crop about 5 years ago of Joe's long cayenne pepper. So I came into pathology through seed, and hopefully I can provide some useful tips for growing healthy seed. This topic can go in a lot of directions, but I'm going to give you a brief overview of how to diagnose disease in your seed crops, how to test your seed to see if it's infected, what treatment options you have, and what regulations you should be aware of, especially if you're selling seed commercially. And many of these topics are very crop specific, and I don't have time to go over them crop by crop, so this will be general, as Jared has already mentioned. So if you have questions about specific crops, we can address those at the end or steer you in the direction of more information. Seedborne pathogens are important for three main reasons. First, infected seed can act as a pathway for a new pathogen being introduced into an area or region where it previously wasn't located. And this happens all the time, and this happened with cucumber seed, which was infected with cucumber green mottle mosaic virus in California about 2 years ago. And this virus occurs in many places around the globe, but it hadn't been in North America yet. So in addition to new pathogens like CGMMV, new pathogen strains and or more virulent strains can also be introduced on seed. And we see this happening with many races of downy mildew and baby leaf spinach production. Secondly, infected seed can act as a significant source of inoculum, which leads to disease outbreaks. And we see this with downy mildew on baby leaf spinach too. Not only are new strains coming in on seed, but existing strains can provide enough inoculum to lead to outbreaks. Seeds, especially with baby leaf spinach, are planted at very high rates per acre, and they're overhead irrigated, and that creates ideal conditions for disease development. So even low levels of infected seed can be damaging in this sort of a situation. And thirdly, seedborne pathogens such as Alternaria brassicicla or Alternaria brassicae, which are pathogens of the crucifer family, can actually impact seed quality by reducing the yield. The picture here shows a germinated turnip seed plated on a nutrient auger to assess seed health. And if you've ever spent time really looking at fungi, you might be able to see that there are a number of different fungi on the seed, and not all of these are pathogenic. There's a lot of microbial life on seed. It'd probably surprise a lot of us. As agriculturists, I think it's fun to point out the work of those before us, and also to point out that seedborne pathogens are not a new phenomenon. Seed pathology, or the study of seedborne pathogens, has been studied formally for 100 years. The USDA APHIS Plant Protection and Quarantine started under the Quarantine Act of 1912 with the explicit goals to protect the nation's agricultural resources from exotic plant pests and diseases, and to prevent the spread of exotic plants within the United States, and thirdly, to certify U.S. agricultural products for export. Paul Niergaard is well known as the father of seed pathology, and the term seed pathology was coined in the 1940s by Paul Niergaard and Mary Noble. You can see them smiling there in the front row. And this study was first used to describe seed testing technology, and this picture was taken back in 1958. So Jared talked a lot about how to think about managing diseases, and I think a useful framework to put this in is the disease triangle, which pathologists use a lot, not just in seed pathology, but in all pathology. And the biotic and abiotic factors that we are looking for when we diagnose, we're looking at these biotic factors when we're trying to diagnose a disease, but these factors don't just cause disease on their own. So if you think of disease as a triangle with 3 required components, a susceptible host, a conducive environment, and a virulent pathogen, when you think about managing diseases, think about reducing the impact of any one of these components. In other words, if you make the side of the triangle smaller, then there's going to be less disease. So for example, Jared gave the idea that if you reduce overhead irrigation, you will significantly decrease your conducive environment. So, here's a smaller triangle with less disease. And then if you switch to a host crop that's less susceptible or has better genetic resistance, you're reducing your disease again. When you think about growing seed crops, these crops are in the ground for a long period of time, and they're exposed to many different environments. So the environmental side of that triangle is really long with seed crops. Some of those environments may be more or less conducive to certain diseases. Here in Western Washington, winters are ideal for the development of certain fungal diseases. These pictures show lightly spot, which is actually the pathogen and disease that I'm working on for my master's thesis. So that is the example I'm using here. But we do have these wonderful dry summers that leave a great window when disease might not advance and develop on the pods, and allows you to kind of bypass some of those environments that would be really conducive to disease. If you have an unusually rainy season and you miss that opportunity, disease can be devastating, especially in these seed crops. The picture on the left shows a field of mustard cover crop, and Jared talked a little bit about this, but they're planted at a much higher density, and fortunately seed crops are usually spaced much further apart to have more airflow and circulation. Jared also mentioned green bridges, and this is a good connection back to that, at least within Western Washington. Many brassica cover crops are grown for seed. They overwinter, and veggies and cover crops brassica can go in at different times of the year. And basically what that means is there are brassicas in the ground year-round in western Washington, and this means that there are plenty of reserves and places for diseases to develop. And this is a continuation of that cycle. So you saw the infected pods on the last slide, and now you can see a picture of an infected seed. And what's interesting with seed-borne pathogens is you also need that conducive environment on the other side when the seed is germinating. So if that seed gets sent off to be grown somewhere that's very dry, maybe you're not going to have as much a problem. If it's sent off to the southeast in a hot, humid, plenty of rain, you might have a bigger problem. So that environment extends to the next set of plants in seed pathology. So even though seed-borne pathogens have kind of solved two sides of the disease triangle by having both a susceptible host and the virulent pathogen kind of all in one package there, they have that added length in the environment. And some pathologists describe a fourth factor of time. There's just much more time when working with seed crops. They can be in the ground for longer, and it has to extend into that next crop planting. So if you suspect disease or you see symptoms in your crops and you're familiar with symptoms and or that plant problem in the field, you might be able to diagnose it yourself. And there are lots of resources out there to help you. Jared mentioned the APS Compendium of Diseases, and these books can be a bit spendy, but they're definitely worth it, especially if you're working with one particular kind of crop. Out here in the Pacific Northwest, Dr. DeToy and a few other researchers from Washington State and Oregon State have a vegetable extension group that has great pictures of diseases, and I'm sure if you look at land grants and extension in your areas, there are similar programs. And APS is a more research-focused website, but they will publish disease notes, especially if new diseases show up in an area. So that's a good place to check and see, is this something new that I'm seeing? Has it been here before? You can check out those at the APS website, the American Phytopathological Society. More often than not, if you're unfamiliar with the plant problem you're seeing in the field, submitting a sample to a diagnostic lab or clinic is a really good option. In the case of many biotic problems, you might run into, you might not have the necessary equipment or expertise to make a proper diagnosis. And again, with these high-risk seed crops that are typically worth more money, getting a proper diagnostic or a proper diagnosis is key. There are many plant clinics and experts throughout the country that can help you with a diagnosis. They have microscopes, tissue culture labs. They can even sample and extract and look at DNA to help you get a good diagnosis. When working with seed-borne pathogens, it can be hard to find conclusive information on whether or not they are seed-borne. There are lists out there, but there's not one central place you can go and see if, is this a seed-borne pathogen or not. And this is why having a good relationship with a diagnostician or a plant clinic is so useful. They can kind of guide you through that process of, is this seed-borne? If I'm selling the seed, is there a quarantine in Idaho about bringing seed that's infected with this pathogen? They can kind of help point you in the right direction and give you more resources. I threw this picture in here of tobacco rattle virus on peony, and it's not necessarily seed-borne, but I thought it's fun to see beautiful disease symptoms instead of just the moldy-looking ones that we typically see here. And I will say one more thing about this before moving on. When you submit a sample, how good and how expedient your sample comes back is directly related to the kind of sample you take. So I'm going to give you a couple tips for submitting a useful sample. And in addition to being tips for submitting a sample, it's just a way of kind of developing a diagnostic mindset so that if you see a problem in the field, you have the tools to kind of start asking questions and figuring out where you're going to, or how you can figure out what the problem that you're observing is. So consider that disease triangle when you're submitting a plant sample. Accurately conveying the problems you're seeing is key. Submission forms that are available from your plant clinic's website may ask crop information that's listed here, the age of the plant, size, cultivar, location of the plant, and the field and the farm, exposure, weather, and just general history of what you've been doing in your field. In addition to that basic information, you can start to think about these 5 different questions. And again, this, if you're diagnosing it yourself, these are useful too. These are the ones that we use here in the lab when we get a sample in from a farmer's field. What was the timing of the symptom symptom development on your plant? Biotic problems don't tend to appear quickly and all at once. So if you wake up one morning and you see something's changed, that usually points to abiotic problems. Is more than one plant species affected? Biotic problems tend to be more host-specific, and so if you see something that's affecting your brassica crops, is it also affecting the brassica weeds nearby? Or if you're looking at spinach, for example, and you see that the brassica weeds are also infected, that might throw up a flag that maybe you're not looking at a biotic problem. You want to observe how the field is affected on a macro level. Is there more disease in lower parts of the field? Is distribution of symptoms sporadic and random in the field, or do symptoms appear to be moving in from one side of the field? It can give you an idea of how things are spreading, or if they're spreading, which will point to different diseases. You'll want to look at how the plant is affected on a micro level. So if you look at the plant, do symptoms appear on newer or older growth? If you have a nutrient deficiency, this tells you a lot. Is this a mobile nutrient or an immobile nutrient deficiency that you're seeing? And the 5th question is, what symptoms or signs can you observe? So what is the actual thing that you're seeing that looks abnormal on your plants? So if you diagnose a disease, or you diagnose a disease with the help of a plant clinic, and it is in fact a seedborne pathogen, you might want to look at finding a lab to test your seed lot. Just because the crop is infected doesn't always mean your seed is infected, so this can save you a lot of time if you don't find that you actually have infected seed. And these seed tests for the health are very specific to the pathogen. They're very specific to the seed crop, and so really if you find yourself with a seedborne disease or seedborne pathogen on your seed, you're going to want to contact a lab to help you through this. And there are 3 labs that have been around for a long time and are well known for working with vegetable crops. If you're working with something like row crops such as corn or soybeans, you probably have more, or you definitely have more options. But otherwise, for vegetable crops, the California Seed and Plant Lab, the Eurofin STA labs in Colorado, and the Iowa State University Seed Science Center and their seed testing laboratory works with more than 300 species of seed. So those are all really great resources. The state departments of agriculture may have seed programs, but not all state departments of ag do, and some have seed testing labs, so that's another avenue you can explore. Again, developing that relationship with your plant clinic to help guide you through this is key. And you can also visit the National Seed Health System website. That's listed here at the bottom bullet. This is a program authorized by the USDA. It's administered in Iowa, and it helps accredit private and public labs throughout the country and acts as a clearinghouse for information. There are multiple ways that seed can be tested for different pathogens. So when you work with these labs, just make sure you know how it's testing and what it's testing for, and be very clear on that. I'm going to talk really briefly about options for organic seed treatments. And again, this is one of those things where it's going to be really specific depending on what you're working with, both crop and pathogen. But in general, here are the tools that work well for organic systems. Hot water treatment has been shown to work really, really well for a lot of different crops. Both bacteria and different fungi respond well to this treatment. Hot water treatment can cause a reduction in vigor over time. So hot water treated seed should be fresh, and it should not be kept longer than 1 season. So if you find that you have a disease in your seed crop, or if you find that you have infected seed, you don't want to treat a giant lot that you're going to be using for years and years. You want to treat it kind of as you're either selling it or as you're using it on your farm, because it can and will reduce vigor over time. Some companies do their own hot water treatment, or they can custom hot water treatment upon grower request. You'll have to ask around for resources if you don't want to do that yourself, or if you want somebody else to do it. There are different ways you can set up home operations for having hot water treatments. And this link here, it's kind of hard to read, I apologize for that, but if you look at the Ohio State University fact sheet on hot water treatment of vegetable seeds by Sally Miller, that's a great resource for figuring out how to do this yourself. And in general, the techniques you want to use are these 4 steps here. You want to warm the seed in water, and what the temperature of that water depends on what the seed you're working with is. You want to heat seed to a set temperature for a set duration of time, and again, that temperature and duration depend. And you want that pathogen in usually circulating water baths. You want to make sure that the water's moving and getting all the seed evenly. Then you want to cool the seed in cooled water and dry rapidly and thoroughly. This technique doesn't always work, or does not work for large seeded vegetable crops, but it's been shown effective for brassicas, carrots, tomatoes, peppers, and even to a lesser degree celery and lettuce and spinach. The next bullet point there you'll see is disinfectants. The example I have is bleach. Bleach can be used to surface disinfect seed as another alternative to hot water. And it will eliminate pathogens on the seed surface, but not if they're underneath the seed coat. So where they are in the seed does matter for some of these treatments. And sodium hypochlorite is allowed on organic farms, and it's often used as a disinfectant in wash water for produce too. And then there are lots of products out there that are considered biological seed treatments, and these are often used in conjunction with priming or pelleting, and they may have potential in some situations for improving seed health, but honestly it seems that in research most of them do not live up to their claim, especially when we have such good options in hot water and in disinfectants. There are a few companies that might be able to help you do a seed treatment, or you could contract with them to clean your seed. Germain's Lab and Ecotech are 2 bigger companies, but again, check with your diagnostic clinic to see if there are other resources out there. And thirdly, or I don't know if this is thirdly, but we want to talk a little bit about quarantines, and this is a little bit dry and less exciting, but it's important to think about if your seed lot is moving around after you harvest it. If you're saving it and putting it on your own or for your own purposes, it's not an issue, but if you're selling your seed, it's important to be aware of quarantines. Quarantines can be international, domestic, at the county level. My advisor pointed out to me recently they can even be sub-county. The county we're in here, Skagit County, only half of that actually has a quarantine for one disease, which I'll talk about here in a minute. So being aware that if you're moving seed into some area with a quarantine, that there are different expectations. Famous examples of quarantine you've all probably heard about, and these are at different levels of regions. Mad cow, scrapian sheep, white rotten onions, rhizomanian sugar beets, and potato cyst nematodes. They're regulated nationally by USDA APHIS, and they're regulated domestically by state departments of agriculture. So for example, if I wanted to learn more about what quarantines there were in Washington State, I'd go to the Washington State Department of Ag website. I'd probably, it wouldn't be super easy to find, chances are, and contacting people to figure that out is often the best way to get clear, up-to-date information about quarantines. And it's important that as seed growers you understand the risk of moving these seeds into areas where there are quarantines, because all quarantines for good reasons. And I think this next slide shows a really good example of a quarantine. So the example I'll give here is the one that I was talking about here in Skagit County, or I should say in Washington. It's the blackleg and black rot on crucifer quarantine. And this is the blackleg or fomalingum and black rot, which is caused by the bacteria Xanthomonas campestris, is in 6 counties in western Washington. And actually blackleg has just recently been extended, I think this was last year, to 20 counties in eastern Washington. And this does not apply for black rot, just the blackleg. And these are zero-tolerance pathogens, as they do not currently exist, at least to our knowledge, in these quarantine zones. And one of the reasons for having such a strict quarantine zone is because these regions where the quarantines are set up are important seed growing areas. So if we don't have the disease here, then we don't have to worry about spreading inoculum or introducing the pathogens on seed into other areas. So it's protected because of the seed production here. And this quarantine bans planting any seed infected with with blackleg and black rot in western Washington, and requires testing of crucifer seeds. And the lots should be tagged, and the tag, you can see that green tag from the Washington State Department of Ag that says that it has been tested and therefore can be planted in the quarantine area. The WSDA, or the Washington State Department of Ag, advises that seed producers become familiar with and follow the regulation for crucifer seeds. Most of the established seed businesses in Washington State have been voluntarily practicing these requirements, and growers in this area are often told to look for these tags on the seed that they're buying to ensure that they're free of blackleg. And if blackleg is found, these fields are destroyed, so that is a high risk for the grower. And as you might be wondering, okay, if this is a voluntary program, how well is it enforced? And that's part of any quarantine, is trying to figure out and do outreach to let people know that if they see these things, this is an important area to keep these pathogens out of. And then lastly, oops, sorry, the slide doesn't look so great here, but if you're shipping or receiving seeds internationally, which many of you might not be doing, but if you are, it's important to consider the importing country's restrictions on seed. And sometimes this means you need a Phytosanitary Certificate. These are issued by the USDA APHIS PPQ. And what these do is they certify the country of origin, that plants are free from quarantine pests and mostly free from injurious pests, and that they meet the entry requirements for that importing country. And APHIS can give these FPCs by visually inspecting the product, or they can do it after inspecting the mother plants while they're still outgrowing in the field. And in some ways that's much easier than submitting your seed to be tested in the lab and taking time to do that. So growing a healthy crop that can be inspected is a great way to show that you don't have disease in your field. So with that, thank you very much for your time, and I think Jared and I can take some questions.
Okay, thank you. Also, if you have general questions about organic agriculture which aren't specifically about diseases and seed production, you're welcome to use the e-extension Ask an Expert service at the link on your screen. So with that, I'll move on to some questions. We've gotten a couple of questions so far, although not so many. So the first question is more about a pest. Can anyone direct me to information on parsnip webworm? It's Pastinella depressaria, and this person is interested in control information in organic production. Do either of you have any information about that pest?
I do not, unfortunately.
Nor do I. Sorry about that.
Okay. Yeah, I did send the person an article that I found with the contact information of someone who seems to be more knowledgeable about that, so hopefully that helped.
Thanks,
let's
Alice.
see. Yeah, no problem. Okay, the next question is part of the problem not having sufficient biological diversity in fields to keep the pathogens in check. I look at the fields in the photos and I see finely tilled beds and acres of the same plant being grown.
That's a good question. I'll jump in first, and Shannon may have some better research-based input on that. I do think that the importance of providing some degree, especially when we're talking about pests, having some degree of diversity in your fields, whether you're talking about having plantings to promote beneficial predatory insects, or having diversity is to be able to kind of create a more complex ecosystem is helpful. And then the other side of that is if you've got smaller patches of plantings, then you have a little less likelihood for a disease to really take off and grow. I think relative to what you see in conventional production and conventional seed production, the scale of production is certainly a lot smaller. The flip side, of course, of kind of diversity in a particular field is along the lines of what Shannon and I both talked about in terms of the green bridges. If you've got diverse planting mixes that are growing over the course of the entire year, you may have the ability for those diseases to persist for longer.
Okay.
Yeah, I think just to add on that, at least here in the Skagit County, when there's this kind of cool element of you want a long crop rotation between seed crops, and not just for disease pressure, but also because you don't want volunteers that could, cross-pollinate your plant. So you see a lot of times that farmers are aware of having longer rotations, and for that reason, but it also works well in the seed production model. But certainly having good, healthy crop rotations is essential to properly managing diseases.
Okay. Is there a way to know if tomato seeds carry tobacco mosaic before planting them? And if they do, how do you treat them?
I think, let's see, I would go check out the National Seed Health System or the International Seed Health, there's an I, ISHI, I forget what the last I stands for. Those websites will have protocols, and I think that they do have one for detecting tobacco mosaic virus on tomato. I haven't worked much with viruses on seed, and so they can be a little trickier to treat as well as detect. So I think taking a look at that ISHI website, which, let me look it up here. If you go to ISHI-veg, that is a good spot to look for that protocol to see if there is a test out there.
Shen, if you wouldn't mind typing that into the chat box, then I can share it with everybody, because I couldn't quite get that just from hearing it. So I think the sound is a little rough. So yeah, meanwhile, I'll ask the next question here.
Okay.
Okay, and if I get that, I'll pass it on to everyone else here. Are dry-seeded crops more likely to carry seed-borne disease compared to wet-seeded? I'm thinking of how long these pods are exposed out there compared to, say, a tomato or pepper when the seeds are protected inside and are harvested when market mature. Anyone have any thoughts about that?
I mean, yeah, Shannon again may have things to chip in on that. My anecdotal understanding is that is true, that you have two things going for you with wet-seeded crops, at least in reference to fungal and bacterial diseases, that they're protected by this thick, fleshy fruit. So the diseases are going to have to either be something that's systemic, that's infecting the entire plant rather than just a surface infection, and if or if it's surface, somehow it's going to have to be able to penetrate far enough to get to the seeds. The other thing is, it's my understanding that the fermentation process that occurs, at least for tomatoes and cucumbers, but many growers apply it to other wet-seeded crops, that process helps reduce some degree of pathogens that may exist on the surface of the seeds.
Okay. Thanks, Shannon, by the way, for that protocol. I just forwarded it to everybody, so you should see that in your chat or question box. Do you have any recommendations for treating seeds that will be stored for a long time, maybe neem oil or other products like diatomaceous earth?
I think, and I could be wrong about this, that the general idea is that, especially with some of these organic treatments, that long-term storage after treating is not always advisable. Would you agree with that, Jared?
That's my understanding. You may have a better idea of the state of the art. I think that the approach is typically what I had mentioned, which is that if you're thinking about seeds for long-term storage, you want to get them very clean and very dry, and then store them in cold conditions. If you're dealing with pests like weevils and borers, things like that, if you're actually able to get the seeds dry enough and have conditions that allow you to store below freezing, you can actually kill many of those pests if they're stored below freezing for long enough. The other thing that I saw once was Jody Lou Smover, who's the scientist and pathologist over at High Mowing Seed, had done a study on seed-borne diseases in their lots of seed, and she found that for many of their seed lots, just the fact of storing their seeds for longer, that over the course, depending on what the disease was, over the course of months and years, that the diseases would the presence of diseases would get to be less and less on the seeds, so that 2-year-old seed may have, depending on the disease, may have actually been clear of a disease that it had when it initially came in. So those protocols, and then what Shannon said, again, sort of like you're bringing it in, you're just going to try and get the seed clean and very dry and store it in cold, dry, safe conditions, and then when you're thinking about treatments like the hot water, you're going to be doing that kind of as it's going out again.
Okay, great. Do you have anything to add, Shannon, or should I move on?
I think just reiterating, you want to treat it before you use it. That's key.
Okay. Question on whether powdery mildew can be transmitted by seed.
Is there a crop in particular that this person's interested in? It depends on the crop to some degree.
Okay, she put lettuce.
Lettuce?
Mm-hmm.
I believe that it can be. So I think that's something to look out for. Was there anything else to the question in terms of?
Nope, that was the question, whether it can be transmitted by seed.
Okay.
Yeah. Okay. Do you have any thoughts on disease management in the production? I know it's a huge question because, you could have an entire webinar on the subject, but on protection of tubers. This is specifically about seeds, Governor, but if you have any advice for a questioner here on.
Yeah, that's a challenge. When we're thinking about tubers or other, asexually propagated crops, so potatoes and garlic, the disease control is a significant challenge, especially when you're talking about viruses. So for many of those, you end up having a system where you are trying to start with something that's very clean, and that either is because it's more and more we're starting to see a movement actually to true seed production in many of those clonally reproduced crops in part for disease management, but many of them are reproduced by initially by tissue culture to try and reduce the amount of pathogens that are on it, and then you just need to grow those crops then in a place where to kind of minimize the amount of pathogens that accumulate on it, and then you end up needing to sort of restart by going back to tissue culture occasionally. Shannon, you may have more detail in terms of the process for that.
Yeah, I really haven't worked much with clonal propagation. I mean, it's a whole different issue in terms of there's so much more that can accumulate in those clonal tubers or roots or whatever.
Were you there when the Seed Savers did their cleanup of their collection? Yeah,
and that involved a lot of very lab-based cleanup of some of those viruses and things where you're actually kind of grafting parts of meristem that you're hoping don't have the pathogen on them yet to newer or to clean stock and things like that. So it's a fairly complicated process to get those cleaned up.
Okay, let's see. This person is having a wet August and is trying to dry rye cereal in an acre in tied bundles in a high tunnel. Is there a better way?
Sorry, can you repeat the question?
Sure. Yeah, okay. This person is having a wet August and he's trying to dry rye cereal, 1 acre's worth, in tied bundles in a high tunnel. Do you know of a better way?
So they're actually making like sheafs of rye with the stalks and the plants. Yeah, I mean, that's a tough one. So you said it was about an acre of crop that was being harvested. I think that.
Yeah, that's what he said.
Yeah. And it sounds like it's at this point it's probably too wet to thresh. That's why it's still on the plants.
Yeah, they're in south central Alaska apparently.
Okay. Yeah. I mean, I think that high tunnels can be an appropriate place to make those attempts to get your seed crops dry when you're dealing with wet weather outside. What you want to be aware of course, is that high tunnels are a source of high humidity. If you're going to do it, I mean, the bundles themselves, you're going to want to just keep them relatively small so that you're maximizing, minimizing the amount of volume in a bundles, because if you're going to have diseases that are going to be in the middle where it's more humid, you're going to want to make sure there's lots of airflow going through your high tunnel, so plenty of fans, and then sort of as early as reasonable, as soon as things are dry enough, you're going to want to try and thresh it, because the seeds are going to be drier than the chaff. And so if as soon as you can thresh it, and then after it's threshed, it's probably still going to be pretty moist, so you're going to want to come back through and maybe put those seeds on screens with airflow coming through those screens, still in the high tunnel, but in a thin layer on the screens to try and get them better dried.
Okay. Let's see. This person says, I've heard that fermenting tomato seeds is essential to preventing disease. Is this true?
Shannon may have more research on this, but that's my understanding, is that not necessarily that it's essential, but that there has been some evidence that the fermentation process, if it's done well, so meaning that if you're fermenting at relatively warm temperatures for a relatively short time, that process will help reduce some of the pathogens in the seed. It depends obviously on which pathogens you're talking about, but that can help. I know that that's my understanding. I apologize, I actually haven't. That's something I've heard secondhand and haven't seen the primary research myself.
Yeah, I'd second that and say it depends on what pathogen, and I've heard that as well, and I'm actually making a note to research what exactly which pathogens it's suggested that those might help with. But generally it seems like it's just a good way to make sure you're cleaning your seed before you start drying it down. You're getting all of that, the placenta of the seed off where maybe something could grow if you didn't get that clean. So I think it's both a matter of the process, and it's just a more efficient way to get your seed clean too.
Okay, well that was the final question, but we still have a little bit of time, so if there's anybody else that has a question, let's just leave them another minute or so time to just type that in. This is your chance, so get a direct answer from the experts. So I'll just leave this open for a moment here. And meanwhile, I just want to mention that our next webinar is coming up on the third Tuesday of next month. And we have quite a few webinars planned for the fall, some of which I listed for the first time yesterday. So if you go to that link on your screen of the upcoming and archived webinars, you can register for all of those. You don't need to register again for any of the webinars in the seed series, because one registration takes care of all of that. So here's the last question here. Oh, there's two. Okay, first one is, how long would you recommend fermenting tomato seeds for?
Okay, so it's going to depend a little bit on the temperatures, but you're going to preferably want it to be relatively warm, so over 70 degrees during fermentation, and then I'm usually looking at somewhere around a 48-hour period, but it's going to depend. The best way to do it is just to sort of check on it and see once you've dissolved the the mucilaginous coat around each seed, then you're there.
Yeah, I don't know about you, Jared, but I've left them too long a couple times and they'll actually start germinating sometimes. I don't know if you've experienced
I've had that happen. I've had it not with tomatoes, but with squash. I've actually left them too long and had them the acids in the fermentation cause the seed coat itself to start to dissolve. So
yeah, you'll see them separate out if you put it in a clear container, or even if it's not in a clear container, you'll see them separate, and the seed will fall to the bottom, and it'll look really clean. And as soon as you see that, you can just kind of rinse off the top.
Okay. This person says she's had lettuce seed that is mostly white, but there are several black seeds are present after she gathers them. So she's not sure whether that was a moisture thing or perhaps indicative of disease. She's in Florida. Just wondering if maybe it could be, cross-pollination or something too. I have no idea. So you have any thoughts about that?
Well, lettuce seed can be both of those colors. So
right.
I think that you'd want to start maybe by planting out the seed separately and seeing if it's different, or but yeah, lettuce seed can be those colors on their own.
Okay. And then final question here. Are either of you familiar with Elaine Ingham and her studies on the soil food web? According to her, in a healthy ecosystem, plants are so coated with beneficial bacteria and fungi that pathogens can't infect the plants.
I am familiar to some extent with Elaine's work, and I do think that first step in any of this is making sure that you have healthy plants and healthy soils, and a healthy system. All of that's going to help reduce the risks of disease. I do personally believe that diseases still occur even in healthy systems, and so whether or not those diseases reach a level where they're problematic for your seed crop, or whether or not those diseases are so serious in terms of their potential, whether they're seedborne and highly virulent, that you need to control them. You may if they're not that serious, and you've got a good system in place with healthy plants and healthy soils, you may not need to be taking other measures to control it.
All right, well, thank you both so much for this webinar. We're running out of time, but I'd like to thank everyone who submitted questions and everyone else who participated. If you just search for eOrganic YouTube channel, you'll find many other webinars that go into even more depth about seed production from many organic seed growers conferences and other webinars that we've done with the Organic Seed Alliance and others. So thanks so much, Jared and Shannon, for presenting this, and to everyone for joining us.